Hello students. Today I am going to discuss the last portion of prokaryotic translation, that is the termination of translation. First you have to know what is termination. Termination means the end. So when the ribosome encounters the stop codons or termination codons on the mRNA, the termination of translation occurs. Now there are three termination codons. Those are UGA, UAG, and UAA. Because this is the termination, so that the ribosomes must be dissociate from the mRNA and the termination machinery. So from this knowledge, we have some idea that there may be some prokaryotic release factor which are basically necessary for this termination and releasing of ribosome from the mRNA. These release factors are classified into two classes. One is class 1 release factor and another one is class 2 release factor. Class 1 release factor are consisting of two release factors. Those are RF1 and RF2, where RF1 recognizes the stop codon UAG and RF2 recognizes UGA stop codon. And the UAA, this stop codon is recognized by both of these release factors. The class 2 release factor is RF3, which is a GTP binding protein, and it stimulates the dissociation of class 1 RF from the ribosome after the release of the polypeptide chain. So basically, translation termination is consisting of three steps. First, the RF1 and RF2 must encounter the stop codon, and second point is that the polypeptide chain must be released from the ribosome, and third one is the dissociation of ribosome subunits from the mRNA. So this is the structure of model of type 1 release factor that is bound at the A site of ribosome, small subunit. And this model illustrates the location of class 1 release factor bound to the ribosome. The P site and E site tRNAs are shown as L-shaped surface, and the GGQ amino acid motif that is involved in the polypeptide hydrolysis is located adjacent to the 3' end of the P site tRNA. This GGQ tripeptide this is the tripeptide that is glycine and glutamine. It is the very much essential tripeptide that hydrolyze the polypeptides from the growing polypeptide from the ribosome. And the SPF peptide anticodon is located adjacent to the anticodon loop of the P site tRNA and in a position that would allow easy to access the stop codon. So this is the mechanism and which is depicted here. Basically, first RF1 is inserted into the A site, where the GGQ tripeptide is located adjacent to the 3' site of the tRNA and of the A site, and it hydrolyzes these polypeptide chain from the tRNA of P site, and this is known as the peptide hydrolysis. And after the hydrolysis of peptide, the class 2 release factor, that is RF3 with GDP, it comes in action. RF3 is the special type of GTP binding protein which has much higher affinity to GDP than GTP. Any other GTP binding protein has the much higher affinity to GTP instead of GDP. The GDP bound RF3 binds to the factor binding site just above to the A site, and it now helps to the GDP now come in action, and it releases the GDP as well as the RF1 from the A site, and the GTP bound RF3 is now acting on the factor binding site of A site, and it provides the energy that is essential for the ribosomal that is essential for the release of the ribosomal release of the release factors or RF1. The ribosome recycling factor now comes in action, and it basically mimics a tRNA. The ribosome recycling factor comes and binds in the A site, and when it binds to the A site, because it is mimicking the tRNA, then elongation factor GTP now comes and sits onto the factor binding site. When the elongation factor G binds at the factor binding site, is basically pushes these E site and P site tRNAs, so that these tRNAs are removed from the ribosomal protein translation machinery. And by this push, this RRF, the ribosome recycling factor, transferred to the P site and the E site, and in the A site the EFG change its conformation, so that the GTP is hydrolyzed, and EFG bound GTP is occupied the A site. Now IF3 comes. This is the initiation factor 3, which I told in the initiation portion of translation, that this is the factor which is very much essential for the dissociation of subunits of ribosome. And when it comes, it replaces the EFG GDP complex, and it binds at the E site of the ribosomal subunits, and thus the 50s and 30s subunits of ribosomes are dissociated from each other with the release of mRNA and ribosome recycling factor. Basically, this is the total process of complete process of termination of translation. Now, if we want to terminate the translation when the termination codon is not encountered, that is, if we want to create a truncated polypeptide, then what will we do? Basically, this is the uh, basically there are some proteins or there are some antibiotic toxins. These are essential for this type of truncated polypeptide formation, and this is very important for your MCQ question that will be asked in maybe asked the role and their target site or the molecular location where they binds. So please go through this table where you can find all the antibiotics. Those can be used to produce a truncated polypeptide in vitro condition. So there is no need to explain this table, and I will discuss definitely the mechanism of this antibiotic, how they produce the truncated polypeptide. Because these antibiotics, so I already told you that all the foreign particles, those are inserted into the translational machinery. They first insert into the A site of the three sites, A, E, and P sites of ribosome. But take one thing in your mind, keep one thing in your mind, that um, only in case of initiation, the initiator tRNA can be inserted into the P site, unless the rest of the tRNAs or anything factors, those are responsible for the translation, they must insert it into the A site of the ribosomal machinery. So here also these antibiotics inserted into the A site can form a covalent bond in between the lone pair electron of the nitrogen and the CO group uh, of the amino acid, last amino acid of the growing polypeptide chain. And thus at the C terminal end, now this antibiotic is covalently linked, so that these antibiotic halts the growing halts the growth of the polypeptide chain, and thus the thus the production of a truncated amino acid polypeptide chain is happening. So this is the main portion where I described how the antibiotics produces the truncated polypeptide. Basically, when an organism is affected by some bacteria or virus, we use some antibiotics. So basically, these antibiotics halts the protein translation machinery of those bacteria and viruses. Those are very much harmful to us, and by this process of halting translation, they basically stops the um, production of functional unit of these bacteria, so that the bacteria cell dies without functioning properly. So basically, this is the mode of action of these antibiotics. And students, this is the end of this chapter. We have already reached the at the end of this chapter. Now it's time to summarize the whole process. Let's see what is the summary. Eventually, one of three termination codons, those are also called the stop codons, become positioned in the A site, and this is the main step. This is the first step of termination initiation. And these codons must be recognized by RF1 and RF2. These codons also do not contain amino acid tRNA complementary to stop amino acid tRNAs. Those are complementary to the stop codon. And instead of these encountering of tRNA, these release factors known as RF1 and RF2 binds at this A site, which recognize the stop codons. The RF1 recognize UA and UAG, whether RF2 recognize UA and UGA. The third release factor is also needed to assist RF1 and RF2 interaction with the ribosome. Thus, either RF1 plus RF3 or RF2 plus RF3 bind depending on the exact termination codon in the A site. If the termination codon contains UAA and UGA, then RF2 will be encountered, and if UA and UAG is there, then must recognition factor release factor is must be the RF1. And RF1 or RF2 bind near the A site, whereas RF3 GTP bind, it can bind everywhere of ribosome. uh, these release factors cause the peptide transferase activity to transfer the polypeptide to a water molecule instead of the amino acid tRNA, effectively cleaving the bond between the polypeptide and tRNA at the P site. And this is the main event by which the growing polypeptide chain is released from the ribosome. The free polypeptide is now leaves the ribosome, followed by the mRNA and free tRNA. This ribosome dissociated into the 5 into the 30th and 50th subunit with the action of IF3, and these are ready to start the translation again. So students, please go through these slides, and please listen this video very carefully. If you have any question, you can ask me anytime you want to ask. So thank you. Please go through this slide.